Hello everyone, welcome, Dr. Jeks here. This presentation is on P.C.R, or the Polymerase Chain reaction. P.C.R is a powerful technique that can make millions or billions of copies of a specific DNA region in a test tube. This technique has revolutionized many fields of biology and medicine, such as genetic testing, forensic analysis, disease diagnosis, and biotechnology. P.C.R was invented in 1983 by American biochemist Kary Mullis, who received the Nobel Prize in Chemistry in 1993 for his discovery. But how does P.C.R work, and what are the applications and limitations of this technique? In this presentation, I will answer these questions and more, using examples and illustrations to help you understand the principles and practice of P.C.R.
Okay, so today we're going to be looking at P.C.R, or the polymerase chain reaction. So the polymerase chain reaction is a laboratory technique which is used to multiply a small sample of DNA. So DNA stands for deoxyribonucleic acid. We all know that every living organism has got its own set of DNA. So sometimes the DNA sample which we need to study may be too small or too little for us to have enough sample to make conclusive results. So we need to make multiple copies of it before we can be able to study it. So sometimes this is very important. Let's say, for example, at a crime scene where you get a very small sample of DNA, like a strand of hair, or sometimes a piece of nail, or some cells, or maybe a drop of blood, or let's say in the cases of rape, some semen. So what we need to do now is we need to take that small available sample of DNA, multiply it, and then be able to generate a large enough number of DNA which you may be able then to analyze. So today we're going to look at the process of DNA multiplication, or photocopying as it were, if you want, which we call polymerization. So this process of DNA polymerization is the one which was taken by scientists, and then they made sure that it's going to be very useful in many scientific investigations. So how does DNA polymerization occur? DNA polymerization is a natural process which occurs in all living organisms. So as we multiply our cells for growth, for repairing of new cells, DNA polymerization takes place. The DNA multiplies into two copies, and the cell will first form a nuclear membrane around the DNA, and then form the cell membrane and form two new cells. So scientists took advantage of this method and commercialized it, and this is now why we have this P.C.R, or the polymerase chain reaction. So what happens during a polymerase chain reaction? Just a few steps. First, we take all the raw materials which we need, the sample from which we're going to extract the DNA. For example, this may be a virus. For example, like we had COVID in the recent past years, where the COVID virus was attacking people. So we take some DNA from the COVID virus, right? Then this is the DNA that we have. Then we put in the reaction mixture in a test tube. We put free DNA bases. You should know by now that DNA has got 4 bases, which are adenine, thymine, guanine, and cytosine. We'll talk about this in another video. You can find it in one of the videos. Then in the same reaction mixture, we've got these comb-like structures, which are the primers. These are what we call primers, DNA primers. These primers are basically a short nucleotide sequence which is going to attach onto the DNA strand. Why do we need primers? Primers are important because DNA polymerase cannot work without a pre-existing chain. So what DNA polymerase needs is a primer, or what you may call a starter molecule, which the DNA polymerase is going to attach to and then continue adding onto a pre-existing strand. So we need a short primer which is going to attach to a known sequence or target sequence of interest, and then this DNA polymerase is going to run in a certain direction. Again, you can see here that we've got these Pac-Man-like structures. If you've played the game Pac-Man, was a classic in the olden days. This pigment is our DNA polymerase. It has been oversimplified so that it may be easy to follow through, okay? It's got this faint green color. You can see it floating in the mixture. So what we will need to do is we're going to repeat a series of steps over and over and over again, and we're going to have our amplified DNA. Another thing which we need to know is that the machine which we are going to use to have or to perform this P.C.R. is called a thermocycler. Thermo meaning heat, cycler meaning to make cycles. So it's going to make several cycles with adjustments of heat, which we will be able to see in a moment. The enzymes that we are going to use this DNA polymerase is derived from a certain bacteria which is very stable to it, which is called TAC. So it's called TAC polymerase, TAC standing for Thermoaquaticus. So this bacteria is very stable compared to it. It actually lives in hot springs, so it is very, very stable with respect to heat. So what will happen here is we are first going to denature the DNA. What is to denature is to change the nature, to change from the original nature to a new nature. So what we will need to do to denature, we are going to increase the temperature in the thermocycler to about 92 degrees. In this range, we're going to use 90 to 96 degrees to work. When we denature, you'll notice that the DNA strands are going to separate and form two strands. It was one double strand, now there are two single strands. After we've denatured the DNA, you can see here that now we've got an open sequence here which the DNA polymerase is going to attach to. But the DNA polymerase is not going to be able to attach because there is no pre-existing sequence. What we want to multiply is this specific target sequence. What do? We find a primer of known bases. Since these bases are known, we are going to use a primer which will attach on one of the ends of this known sequence. And then we reduce the temperature to about 50 to 65 degrees. So when we reduce the temperature, the primers are going to attach to the end of the sequence, which we call annealing. The DNA strands have already been separated. So you can see here that the primers have attached. Then what happens next now is that we increase the temperature. You see now where the thermocycling is happening. First we increase to about 90 to 96 degrees for denaturing. Then we decrease the temperature to about 50 to 65 so that the primers can attach. Then we are now increasing the temperature to about 70 to 74 so that the DNA polymerase can work. So you can see the DNA polymerase running in a certain way. Now take note that the DNA polymerase only works in a certain direction. The DNA strands are so specific that the DNA is going to run in a specific direction. DNA replication happens in a specific direction. We say that this direction is a 5' to 3' direction. Okay, we'll talk about the structure of DNA in another video. So you notice that this DNA is going to run in a 5' to 3' direction. What eventually happens is that the DNA is going to be double stranded according to this. So here we're just representing using one for ease of understanding, so for simplicity purposes only. Right, so synthesis starts at the primer. DNA polymerase runs in the 5' to 3' direction, in the 5' to 3' direction on this other one, and forms how many new strands? One, the original, second, a daughter template, another one, the third, the original, fourth one, a daughter template. Again, we're going to increase our temperature to about 96 degrees Celsius for denaturation. You see now we have 4. Again, we're going to repeat the primer annealing. The primers are always in the reaction mixture. If they run out, we need to add some more. So we've annealed or attached some primers. Again, we are going to repeat this process, and the DNA polymerase, after it is finished the adding, it's going to detach from the molecules. Take note that because DNA polymerase is an enzyme, it is going to be available to participate in another reaction. Why? Enzymes are biological molecules which speed up the rate of chemical reactions. They themselves do not get used up in the reaction. So they are better than chemical catalysts because they're going to be available to participate in subsequent reactions. So you notice that here, our DNA polymerase is very efficient and it is now free in the mixture, ready for another reaction. Because we also said that the DNA polymerase is tough, it's going to withstand these high temperatures. We are going to denature, again, attach some primers and run polymerization. So take note also that this polymerization is happening after a certain amount of time, reattachment, polymerization, denaturation. These cycles keep happening over and over again. Take note of the fact that as this DNA polymerization is taking place, the number of DNA molecules, single-stranded molecules that we're talking about, is increasing not only at an arithmetic rate, but at an exponential rate. What do I mean? In the beginning, we had 2 strands, right? The 2 original strands. After the first cycle, we had 2 strands, the original plus 2 daughter strands, which means we had 4. Okay? So after the first one, we had 4. Again, these 2 daughter strands are going to multiply and produce 2 each. So after the second one, we had 8 strands, right? After the third one, so the first we produced 2 daughter strands. Originally we had 1 strand. After the first cycle, we had 2 strands. Then after the second cycle, we had 4. After the third cycle, we had 8. After the first, second, third, after the fourth cycle, we had 16. Now how many are there? 2, 4, 6, 8, 10, 12, 14, 16 right here. Right? We denature again. Then we anneal some primers. We allow DNA polymerase to take place. Now they are 32. You see that at each cycle they are increasing exponentially with the exponent of 2. So the first one, 2 to the power 0, which gives us 1. The first one, 2 to the power 1. The second one, 2 to the power 1, 2 to the power 2, 2 to the power 3, 2 to the power 4, and 2 to the power 5. Right. So you see that as the exponents are increasing, the number of molecules at each subsequent cycle, the number of molecules are increasing. And it means that just a few cycles we have a large amount of DNA. For example, if we run this process for just 10 cycles, it means we have 2 to the power 10, which gives us 2 to the power 10 is equals to, as you guessed, your answer is correct. So it means that this process continues and then will produce a large amount of DNA. Let me just run a few cycles here and you'll notice that we have a large amount of DNA in the reaction mixture. I'm continuing running these simulations. You can see here that the DNA polymerase, as the amount of molecules is increasing, the reaction mixture is getting crowded. Why? Because of the DNA. I anneal some primers, polymerize, denature, anneal primers, polymerize, denature, anneal primers, polymerize, denature, and the cycle continues until we want it to stop. You see that now we've got, I don't know how many cycles we've ran, and the reaction mixture now is very, very congested with our molecules. You might wonder, what is the use of this polymerase chain reaction? PCR are used in forensic science, in research and genetics, in medicine, can be used in many different ways. In forensic science, it can be used as a diagnostic tool. It can be used in genetic fingerprinting. It can be used in identifying criminals from a large population, can be used in paternity tests, in research and genetics, can be used to compare genome of two organisms in genomic studies, to produce phylogenetic trees, to analyze gene expression, for gene mapping, finding the location of a gene, can be used in medicine to test for genetic mutations, to monitor genes in gene therapy, to detect disease-causing genes in parents, so that, for example, maybe you may want to eliminate the genes during gene therapy, or you may just know the likelihood of a certain genetic disease being passed on to the offspring.
I hope you enjoyed this video and learned something new. If you did, please give it a thumbs up and subscribe to my channel for more awesome content. And don't forget to leave a comment below. And let me know what you think about P.C.R. Do you have any questions or suggestions?